Hello and welcome to this tutorial on DNA, which stands for deoxyribonucleic acid. I wrote it out ahead of time to spare you the pain of watching me spell this in real time. So let's get started with the structure of the DNA, which is the basic unit of heredity. DNA is our basic unit of heredity. Let me have that here for you. Some important comments that might help you. Right. So as I mentioned, it's a basic unit of heredity. This is something what codes for all of our genetic information, or I would say this is something that really makes us us. It gives us our own unique identity. Let me just give you a little background on the discovery of the DNA. This was first discovered in the year 1953 by the two scientists, Watson and Crick. This happened to be a really amazing discovery in the mankind. The journal, which was actually published by Watson and Crick, was just a page and a half long, which contained almost all the information about this unique biomolecule, which is responsible for giving us our own unique blueprint. Anyway, so let me just tell you more about the structure of the DNA. So as you notice, I have this picture right in front of you, which gives us the insight of the structure of the DNA molecule. So I will have some important comments for you over here. The very first one being the double-stranded molecule. As you can see in the picture over here, DNA is a double-stranded helical molecule, which means there are two strands which run parallel to each other. So let me just select different colors for you so that I can probably explain you which two strands I'm actually talking about. Okay, so I have this first strand over here, and I have another strand which runs parallel to the first one. Now these are not just parallel to each other, but then these are rather referred to as anti-parallel DNA strands. So what exactly is meant by this anti-parallel strands? Now coming back to this picture, if you look at the first strand of the DNA, I'm talking about the yellow one, that starts from the 5' end and it ends at the 3' end. However, if you look at the other strand, which I've shown over here in red, that starts from the 3' end and it terminates at the 5' end, which means these are definitely parallel to each other, but then just because of this opposite orientation, they are preferred being called as anti-parallel strands of the DNA molecule. Okay, what else is present in the structure of the DNA? So this was about, the first 2 points were just about the overview of the DNA molecule. Now if we go into the details, I have another important point for you, which is the nitrogenous bases. So this is nitrogenous bases that I'm talking about over here. Now what are these nitrogenous bases? These nitrogenous bases are the ones which are present in the two strands of the DNA, and the arrangement of which gives us our own unique genetic information. Let me explain you this in a much better way over here. So these nitrogenous bases are classified into two categories, where we have the very first one, which is purines, and the other one being pyrimidines. Okay, these are just the two categories that we have for the nitrogenous bases, which in turn is divided, purine is in turn divided into adenine, and we have another one which is guanine. Similarly, for pyrimidines, again we have these two specific ones, where I have thymine, and another one which is cytosine. Alright, so together we have A, T, G, C, which are the four important nitrogenous bases that together make up our genetic information. So it's just the arrangement of these ATGC on this sugar phosphate backbone of the DNA molecule, which gives us our genetic code. So let me just tell you one more thing about the structure of the DNA, which, as I just mentioned a couple of minutes back, as the backbone of the DNA. So backbone of the DNA is nothing but these two strands which are intertwined to each other in a helical manner, which constitute the skeletal framework of the DNA molecule. So this backbone is referred to as the sugar phosphate backbone. Reason, because this is made up of sugars in the phosphate. So sugar, I have a specific sugar over here which is known as the ribose sugar, and that is completely unique to this DNA molecule. It's not ribose, it's deoxyribose, which if you notice over here, I mentioned right at the beginning of the tutorial. Deoxyribose sugar, and the other component of the backbone of the DNA is the phosphate group. Now these two entities over here, they are present in an alternating fashion where we have one sugar, one phosphate, another sugar, another phosphate, which are linked to each other together, forming the backbone of this molecule. Alright, so another important aspect of this DNA molecule, if we look at the structure over here, these two strands of the DNA are separated by a specific distance. We have the specific length which is present between these two strands of the DNA, and that is 10 angstrom. So that's the distance which separates the two strands of the DNA. And this happens to be a very important, unique identity for the DNA molecule. So this diameter is 10 angstrom for most of the eukaryotes, but then this might fluctuate for some of the prokaryotic species like the bacteria, or even the other smaller organisms like the parasites, or maybe Plasmodium, or some other unicellular organisms. But then yes, for most of the eukaryotic organisms where we, the human beings, belong, we have the 10 angstrom distance present between the two strands of the DNA molecule. So just to give you a quick recap about the basic structure of the DNA molecule, it's a double helical molecule where we have two strands of the DNA which are not parallel, but then these are anti-parallel to each other. And I guess now we know why these strands are referred to as anti-parallel, just because of the 3' and the 5' orientation of the molecules. And then also we have nitrogenous bases as again one of the important components of the DNA molecule, where we have purines and the pyrimidines, purines which are adenine and guanine, and pyrimidines which are thymine and cytosine. One quick thing which I forgot to mention about over here is the complementarity. So adenine is complementary to thymine and guanine is complementary to cytosine, which means on the strand of the DNA, let's assume this is one strand of the DNA, if I have A, T, C and C, then on the complementary strand of the DNA over here, we will have the complementary nitrogenous bases, where A would be complementary to T, T would be complementary to A, C would be complementary to G, and C again would be complementary to G. So present between adenine and thymine is a hydrogen bond which actually connects these two nitrogenous bases together, and A and T are connected with two hydrogen bonds over here, again two hydrogen bonds, but then between cytosine and guanine we have, instead of two, we have the presence of one more hydrogen bond. So yeah, overall 3 hydrogen bonds present between guanine and cytosine, and this is something which is common across the border. Like every single molecule, every single nucleic acid will have this kind of arrangement where A will always pair with T with 2 hydrogen bonds, G will always pair with C with 3 hydrogen bonds, and this is something which is conserved, and because of this DNA molecule is referred to as a very conserved molecule, just because of the unique arrangement of these ATGC, which are connected to each other by this complementary nature. So that's all for today's tutorial. We'll come back with some more information on the structure of the DNA. So far, this is what it is. Thanks for watching this video. See you next time.